So how can a single encounter with a virus lead to such deep and lasting problems across the body's most vital systems? It's a huge puzzle, right? And the answer doesn't point to just one single failure, but to something much more sophisticated, a coordinated attack. In this explainer, we're going to break down exactly how that works.
The entire story really hinges on these two tiny fragments, located just 49 amino acids apart from each other. Think of them as a team. One is the payload, which carries the blueprint for an autoimmune attack. The other is the trigger, a kind of facilitator that takes down defenses and opens up doors. And together, they launch this incredibly precise two-pronged attack on the host.
Okay, to really get this, we're going to deconstruct their strategy piece by piece. First, we'll dig into that autoimmune payload, what it is and what it targets. Then we'll look at the functional trigger that makes the whole attack possible. After that, we'll see how they work together to create such devastating consequences. And that's going to lead us to a final and really crucial question. All right, let's dive into our first section, the autoimmune payload. This is all about that RLFRK fragment. You can think of this piece as the blueprint for destruction. It's the bit of code that's responsible for teaching the immune system to attack the body's own tissues.
So, what is this RLFRK thing anyway? Well, it's just a tiny sequence of 5 amino acids, but what makes it so dangerous is its location. It's right there on the surface, totally exposed, which means it's incredibly easy for immune cells to see it. And here's the key part. Studies have actually confirmed that this exact fragment is floating around in the bloodstream. That means it's biologically available to kickstart the immune system on a body-wide level.
Now, to understand why a 5 amino acid sequence is such a big deal, we have to talk about a concept called T-cell cross-reactivity. Some people call it the rule of 5. See, antibodies need a perfect lock and key fit to recognize something. But T-cells, they're way more flexible. They only need to spot a core sequence of 5 shared amino acids to sound the alarm. And since RLFRK is exactly 5 amino acids long, it's the perfect trigger for this kind of molecular mimicry, right out of the gate.
And that right there is the consequence. Because of this rule of 5, any of our own human proteins that just happen to have that same 5 amino acid sequence are now at risk. They can be mistaken for a piece of a virus, leading to a massive case of friendly fire. In other words, autoimmunity. This, of course, leads to the most important question. What vital parts of our own body are actually in the crosshairs of this autoimmune attack?
The first target is an enzyme called PPIP5K2. Think of this as the power source for our body's main antiviral alarm system, the Type I Interferon system. It's what provides the juice needed to flip the switch. So when an autoimmune attack hits PPIP5K2, it basically cuts the power. And the result? Our first line of defense just collapses, even when a virus is clearly present.
But the attack doesn't stop there. This second target is a protein called POMGNT1, and this is where things get structural. This protein is essential for making sure other proteins, especially in the brain and muscles, fold correctly. When it's attacked, proteins start to misfold, risking a total structural collapse. And to give you an idea of just how serious this is, the failure of this exact mechanism is what causes very severe genetic disorders like muscle-eye brain disease.
The third major target is a protein called 10M4. This one plays a huge role in myelination, the process of insulating our nerve fibers so they can fire off signals super fast. When T cells go after 10M4, it can lead to demyelination. This is like stripping the insulation off a wire. It causes nerve signals to slow way down and is linked to some really serious neurological disorders.
Okay, so that's the payload, the ammunition for the autoimmune attack. But, ammunition is pretty useless without a trigger, and that brings us to our second section, the functional trigger. Here, we're going to look at its partner in crime, the RGD fragment, which is both a saboteur and a facilitator. The RGD fragment works in a totally different way. It's not trying to get noticed by the immune system. Instead, it acts like a tool. It's a master of disguise that latches onto our own integrin receptors, which are basically these sophisticated signaling hubs on our cells, and it hijacks them to send disruptive signals all over the body.
And the first disruptive signal it sends is a really potent one. When RGD binds, it basically rips a biological safety leash off a powerful molecule called TGF-β. Once that's unleashed, TGF-β acts like a widespread immunosuppressant. And get this, it shuts down that exact same Type I Interferon pathway we were just talking about. This basically blinds the host's innate immune system, creating the perfect environment for a virus to go wild.
Next up, this RGD integrin interaction does something even more direct. It physically starts to dismantle the blood-brain barrier. This causes the barrier to leak, compromising the integrity of this critical fortress. So the main takeaway here is that RGD opens a direct hallway for inflammatory molecules and those autoimmune cells we talked about to just march right into the central nervous system.
And finally, RGD acts like a fire accelerant. It triggers a cascade that ramps up the production of IL-6, which is a major inflammatory molecule. This IL-6 then acts as the primary signal to create more pathogenic Th17 cells, and these are the specialized troops that are notorious for driving severe autoimmune diseases like MS and lupus.
So we've seen the payload, and we've seen the trigger. And yeah, on their own, they're dangerous. But now, let's put it all together. In this third section, we'll look at their synergy and the consequences. This is where you really see the devastating brilliance of this coordinated attack. Let's look back at the attack on that antiviral alarm system. It's a double hit for redundancy. RGD jams a signal by releasing TGF-β. That's like cutting the alarm wire. And at the exact same time, the RLFRK-driven autoimmunity is destroying the power source, that PIP5K2 enzyme. So with the wire cut and the battery destroyed, that alarm is never going off. That guarantees the virus has plenty of time to replicate.
And this synergy is even more obvious when we look at the brain. RGD is the key to the fortress, right? It breaches the blood-brain barrier. At the same time, RLFK is providing the troops, those autoimmune cells that are specifically trained to attack neurological targets. So the attack literally provides both the access and the specialized forces needed for a full-on, targeted invasion of the central nervous system.
And this whole thing isn't happening in a vacuum. This destructive cycle gets amplified by our own body's systems. Key signaling pathways get hijacked, fueling even more inflammation and dangerous clotting. The body's natural brakes on the immune system just fail. And worst of all, this whole process provides a growth engine to create these sort of immortal, hyper-aggressive T cells that the body just can't get under control.
And as the source material puts it so clearly, when these natural self-extinguishing mechanisms are compromised, the pathology driven by RGD and RLFRK spirals into a vicious cycle of chronic destructive autoimmunity.
So understanding this whole mechanism, this really intricate, coordinated attack, it doesn't just explain the how. It forces us to ask a much deeper, more fundamental why. And that brings us to our final section, and a really profound question about the cost of survival itself. So, to sum it up, we have the facilitator, RGD, which is opening the doors and shutting down defenses. And then we have the ammunition, RLFRK, providing that self-targeting blueprint. And this all leaves us with the central, challenging question posed by this research. When our body launches an incredibly strong immune response to clear a threat, is it forced, by default, to risk destroying, through this molecular mimicry, the exact same machinery that regulates our most critical and fragile neurodevelopmental pathways? It is a really sobering thought, isn't it, about the delicate balancing act our immune system has to perform, and the potential hidden cost of survival.